And good morning, or good afternoon. It's asynchronous. Good day. Hi, everybody. Hey, so it's Friday. I'm putting 2 lectures up today, and I'm looking at the syllabus, realizing, I was supposed to cover chapter 3 and 4 this week. Oopsie. We'll pick up and see where we go. What that may mean is that the test, rather than being on Friday of the 18th, we may push it back. We'll just see where things go. And frankly, I haven't figured out how we're going to do the test yet anyway. But hope you're doing well. Apparently, cases are starting to crawl up number-wise on the campus. But we're doing our best, I'm sure. And it's not bacterial, which is too bad. Probably could have treated it with an antibiotic. But it could have been a much nastier infection if it's bacterial. All right, so when we last left, we're talking about some of the tools and mechanisms for classification and just general kinds of cell biology stuff. This is our example of a dichotomous key in which we sort of work our way through this tree with these different branches and doing tests and visual observation. And one of the points that about this time in lab, we'd be working with microscopes, and it's a bit tedious. Using microscopes takes some practice, so being able to have a virtual microscope isn't necessarily a bad thing, but it is important that we talk a little bit about different kinds of microscopy and microscopes. What you're looking at here in this set of panels is all light micrographs, and meaning that this is taken with a light microscope. And so we can get up to about, 1,000 times the 1,000x, in terms of magnification, which is good enough to be able to see bacteria at a moderate level. You don't get great detail, and there's a lot of things you can't see, but you can see with stains, and you can see the different kinds of shapes, and you can also see where they have either the various flagella or there are different kinds of modal mechanisms. And so the light microscope is a very important tool. Another tool that is also critical to a lot of basic research is the electron microscope. And this is an example of looking at a comparison between a light microscope and an electron microscope. Now, Leeuwenhoek and Robert Hooke, they developed the very first microscopes. These were actually a set of lenses that were just set up in an old frame, and the sample was on a little thumb screw that you could kind of pull in and out, and you could very carefully focus it. They didn't have great magnification. Certainly magnifying glasses had been developed even in the 18th century, but this is able to stack things up. Now, one of the things that isn't necessarily apparent is that the wavelength of the light is a critical component to the degree of resolution. And you can think of this perhaps thinking about TVs that you might have owned in the past. In that resolution is a term we use in the microscopy world for the ability to distinguish between, where's my pictures, 2 points. Beep, beep. And how close can 2 things be that you can still distinguish them as 2 points? And because things aren't necessarily touching, they may be very close. The resolution is the ability to see fine detail. What is the limit of your resolution? And I'm thinking with TV sets, if you've got an old-fashioned TV that's not high def, and maybe like 720 or even older, 720p, that's the pixels and that's the degree of resolution. There's a really big difference when you crank up to a high-def TV. All of a sudden, you can see the pores on people's faces, and you can see their pimples, and you can see the stitching in the costumes. That is an example of increased resolution. And, you can watch a low-def version of a film and still get lots of information. But if you want to see the detailed work on Doctor Strange's costume, man, you go to high def and all of a sudden it's really a different world. And so the resolution is a critical point. Now, light microscopes are limited in their resolution to probably around 1 micron, 1/1,000th of a meter. Teeny, but still, that's the limit because the wavelengths of light are such that they can't actually resolve things any finer than that. And so that means that with a light microscope, we can see a cell pretty easily. We can make out things inside the cell somewhat, but particularly detail inside the cell, just isn't going to be possible. So you can see a mitochondria, but you can't see the membrane inside the mitochondria. Now, we have stains that allow us to visualize things in a slightly more detailed way, and these stains are really, can be quite impressive. Another tool that is often used is an antibody that will bind to a specific protein, and then you have the antibody label. And a number of tools have been invented. One of the things is that the closer the light is to blue, the shorter the wavelength is, and so you can get a little bit better resolution from you looking at blue light versus red light. But our light microscopes, typically, when we're using them, is using just a combination of wavelengths. So there is a limit to how much resolution we can get. Now, just see, looking at the diagram here, we've got a standard light microscope. Let me get my little pointer up. Beep. So we have a standard light microscope in which we've got the light source is going through a series of prisms, and we then have the light coming up through the sample and then into the objective, and then up into the eyeball, and then up into the eyepiece, and then into your eyeball. So you can focus things, you can tighten the beam of light, but this is how it basically works. And in fact, it is based on the same principle that von Leeuwenhoek and Robert Hooke had back in the early days of microscopy. So I will say, even within light microscopes, there's a really broad range. You can have sort of low-def microscopes that are not very expensive and maybe fairly rugged, but you get into multiple hundreds of thousands of dollars microscopes, and wow, the difference is really quite amazing. So light microscopes have a range of quality, and that a really expensive light microscope is still a absolute thing of beauty that is really a lot of fun to work around with. There can be ways of polarizing light in which you can have the light going through a series of polarizing filters, kind of like sunglasses that you might use, and that can interact with material in the sample and change the polarity, and it gives you a sort of black and white kind of image of light that is polarized in one way, can go through the sample, and then if it hits stuff, it gets depolarized, and it's scattered, and it will end up being black or darker. And so you can get a finer level of resolution and sort of almost like a stain with things like what's called phase contrast microscopy. You maybe have seen something called differential interference contrast microscopy, DIC, also known as Nomarski. When you're looking at these light micrograph images that have a 3D look to them, that's actually a lot of fun for samples based on the thickness. The light is able to travel through in different ways and polarize in different ways, and you can kind of shade things where you can have the polarization shade around, and you can get quite a bit of detail there too. But in order to really get the high magnification, to be able to see things like the membranes within the organelles and to be able to see the attachments of flagella, you have to turn to this guy, the electron microscope. Now, electron microscopes come in two different kinds of styles. The one we're looking at right here is something called a transmission electron microscope. It's known as a TEM. And a transmission electron microscope is taking a high voltage beam, and it generates a stream of electrons, which are much smaller than photons and their wavelengths. And it then goes through a series of magnetic lenses that are able to focus this thing, and you end up having the specimen, and then you have the actual screen down here is a fluorescent screen with a camera, and that's what you're looking at. Now, the processing of a specimen for particularly transmission electron microscope is way more difficult. For light microscopes, you can take some bacteria, spread it out on a microscope slide, you're good to go. For transmission electron microscopy, you have to have the sample has to be, or is going to be completely dead. It has to be dehydrated, so there's no water. It has to be coated and stained with materials that are electron opaque, and these are typically heavy metals. They're rather nasty and toxic, and they require a fair amount of different stains and things. Additionally, the sample has to be phenomenally thin, or the electrons can't pass through at all. And so what you end up doing is taking your sample, embedding it in epoxy, and then taking thin sections using a diamond-coated knife. And the thin sections are so thin that they actually are able to reflect light in an interesting way and generate different colors. And you go for what's called a silver section, which is a very, very thin section, thinner than an individual cell. Often microscopists will use a baby's eyelash glued to a little wooden stick to manipulate things around. And so you've got the samples sitting on these little metal grids. Takes a fair amount of time to prepare them. You then put them in the electron microscope, and you can begin to see what's going on. Now, one of the things is that the material in the electron microscope is completely dead, so you can't watch any kind of living process. There are some limitations to it, but this is how, with the transmission electron microscope, you can get down to the spot where you're not quite visualizing molecules, but you're able to see things like the phospholipid bilayer. You can see the chromosomes condensed, and you can make a tremendous amount of detail. And this has really advanced the understanding of how cells work and cell organization. Now, another kind of microscope that is pretty neat, and we're looking at a figure. Back to it. There. This is an example of a microscope view using an electron beam, but it's called a scanning electron microscope. And what a scanner does is, rather than going through the sample, it bounces off the surface. And so the sample prep is a lot easier. You take your sample, you put it into a machine that will coat it with a very thin layer, sort of an atomized atomic layer of gold or other kinds of metals, and you put this into the scanning electron microscope. So you're looking at the surface of things, and that gives you a really cool three-dimensional view. And you can see here that you can pick up a lot of detail of the bumpiness of the surface and all kinds of stuff. And again, the samples are completely dead, so you can't look at a living process, but the sample prep is a lot easier. You don't get into a cell or whatever it is you're looking at the surface of it. So a scanning electron microscope is another really useful tool. So we got transmission electron microscopes, TEMs, and scanning electron microscopes, and SEM. Now, some questions to think about. You can look in your text for more info, as it says. The limitations of light microscopes, that is primarily the wavelength of light is such that there's a limit to how much resolution you can get. Electron microscopes get around these problems by using electrons and not photons. Fluorescence microscopy, this is the tool in which we primarily are using an antibody and then label it with a fluor. And fluorescence microscopy allows you to visualize very specific spots in the cell based on the antibody. And the fluorescence is quite beautiful to look at as well. The contributions of electron microscopes, I'll let you look that up, but primarily the fine structure of a cell and understanding the subcellular components of a cell. And so we just went through the transmission and scanning electron microscopes. Good. All right. Now, this is sort of a catch-all bit here, but we've been talking a little bit about the individual bacteria. And it's easy to think of bacteria as working as independent cells. And that complex organisms, such as myself and yourself, we've got cells that are arranged in tissues, and then these tissues are arranged in organs, and then we have organ systems. And so we've got a lot of cellular interaction. And of course, you've had anatomy and physiology, and you know about the fact that cells can lay down in sheets, and cells can form a variety of different kinds of structures in a complex organism such as ourselves. And with bacteria, it was believed that they were functioning pretty much as independent little organisms, that they didn't have the ability to actually communicate within a community. So cells that are in a multicellular organism, such as ourselves, have to have mechanisms by which we can communicate, and this is done by hormone secretion or cell-cell contact. Certainly the nervous system is, in advanced eukaryotes in animals, is a really good way. Plants, though, don't have a nervous system, but they've got a lot of cell-cell communication. Hormones are being put out by one cell, there's receptors on another cell, and you get all kinds of really cool cytoplasmic activity that says, okay, this is where you are in a tissue, or this is what one cell is saying, how are you going to respond? But we now know that really all cells require the ability to coordinate activities, and this includes bacteria. And it actually has some pretty important medical consequences. And one of the ways that we think about bacteria responding to a change in their environment and able to sense and, in fact, spread that message is a phenomenon known as quorum sensing. Quorum being a required number for some sort of event. Meetings often have to have a quorum. In this case, we've got a large number of bacteria forming a community, and then they have the ability to actually send out signals, and we're only just still understanding some of these signals they can send out. So one of the things that bacteria will do is that a guy that might be near a nutrient source can send out a chemical that's known as an autoinducer. And it's a signaling molecule that is able to generate a signal to other bacteria. They can begin moving around and coordinate to begin, in fact, forming various sheets and different size things. One of the primary autoinducers that was very early discovered was a derivative of an amino acid known as homoserine lactone. And what this allows the bacteria to do is to generate a sheet of bacteria that are essentially talking to each other and communicating. And this is now known as a biofilm. And biofilms, on one hand, sound kind of nice. Oh, a biofilm. Maybe we can use this as a saran wrap. But no, bacterial biofilms, they're not so good. I'm standing in the way here. I'll move over here. Bacteria biofilms are, in fact, a real problem. Bacteria biofilms are sheets of bacteria that are actually able to coordinate some of their interactions. So it's not a single bacterium. And they can embed themselves in material. They can begin secreting material to embed themselves in it, make it harder for them to be washed off. Biofilms end up being a real significant problem when we're thinking about things like a hip replacement or a knee replacement. And that, if you've ever seen a hip replacement procedure or know of this, they have the ball of the hip is absolutely, of the femur, is cut off. And then something that looks like a trailer hitch with about 6 to 8 inches worth of spike, all made of titanium, is embedded into the bone. And then that's fastened back in. I mean, it's a pretty vigorous sort of surgery going on there. This is now sealed back up in the body. If you've got any kind of bacteria that get caught in that part where you're sinking in, or the part where the ball is, they can begin to form biofilms, and they can be very difficult to eliminate with antibiotics. And in some cases, the bacteria infection becomes so severe that another surgery is required to actually pull that joint, that artificial joint out and clean the whole thing out and replace it, and that's actually quite serious. So biofilms are a system by which bacteria can interact with each other, and it makes them that much more difficult to remove. All right. So we're approaching Labor Day weekend. As I've said in one of my classes, at Texas A&M, school didn't start until after Labor Day anyway, so I don't feel like we were behind. We're just now getting started. But we will pick up working on the next chapter here. I'm probably going to do the lecture on Monday, and we'll drop it then and see how we go from there. So here's a summary of our discussion of quorum sensing, is that we've got individual bacteria here in low concentration, and they're sending out signaling molecules, and we then begin to get bacteria sort of moving together. They can modify their metabolism, and it's essentially a way of saying, I am here, and then you get more bacteria forming, and this results in a sheet that can share resources and actually kind of serve as a community protection. So slippery rocks on film, certainly, or slippery films on rocks, but also for different kinds of accumulations, say in a artery and on material within the body after surgeries and things. So this is important because we get these large populations of bacteria. They're able to shield themselves from the immune system, and this can become a pretty serious problem. So things like staph infections on catheters and implants, one of the things that you've been doing any clinical work or been in the hospital setting, that people that have a catheter, they're just really on their way to getting a staph infection. It's not something that you can easily prevent. That inserting catheters and things is always going to have a risk of infection, and the longer it stays in there, the more likely infection is. So bacteria, while they are single cells, they actually have a lot of stuff going on, and they're able to carry out some rather elaborate interactions that weren't always understood. And so we've got then a variety of tools by which we can begin to separate out bacteria and identify them. And we'll be getting into the tools of bacterial identification in Chapter 4 on Monday. Hey, thank you for your attention. We'll figure out the next steps here. Next week is a new week, and I'm getting a better hang of my lecturing. Give me a holler if you have any questions or if you're concerned about things. Otherwise, I will see you here next week. Bye.